สวัสดีครับท่านผู้ชมทางบ้านทุกท่านครับขอต้อนรับทุกท่านเข้าสู่การประชุมวิชาการวัคซีนแห่งชาติครั้งที่10วันที่2ภาคบ่ายผมนายวรวิทย์บุญญาธิสถานพิธีกรดำเนินรายการในช่วงบ่ายของวันนี้ครับสำหรับท่านที่กำลังรับชมไลฟ์สตรีมมิ่งในวันนี้ณเวลานี้นะครับเป็นห้อง N ครับห้อง N ไลฟ์สตรีมมิ่งนะครับสำหรับการเปิดคลิปวิดีโอเมื่อสักครู่นะครับจะเป็น Introduction ในการเข้ารับชม Virtual Exhibition และ Walk Throughการประชุมวิชาการวัคซีนแห่งชาติครั้งที่10ครับในงานเสวนาช่วงบ่ายในวันนี้จะเริ่มต้นด้วยการเสวนาภาคภาษาอังกฤษครับโดยวิทยากรชาวต่างชาติซึ่งมาจากบริษัทผู้พัฒนาวัคซีนชั้นนำของโลกโดยจากนี้จะขอนำทุกท่านเข้าสู่การเสวนาในรูปแบบภาษาอังกฤษครับ Distinguished guests, honorable speakers, ladies and gentlemen, welcome to the 10th National Vaccine Conference which will be held online from March 16 to 18, 2022. Allow me to begin by inviting you to an international session which will be run fully in English without interpretation. So, if you have a question for the speakers, please insert it into the chat box and we will forward it to the moderator. My name is Mr. Warawit Bunyathisathan, will serve as your MC during this afternoon session. Let me start introducing you to our standing session number 3. We will focus on update COVID vaccine data on variant of concern, efficacy, safety, and effectiveness from real-world evidence. Today, we have 6 keynote speakers from around the world who are vaccine development specialists. They are all global leaders in the production and delivery of COVID-19 vaccines. I would now like to turn the floor over to all session executive moderators, Dr. Sunet Chinkit Mongkol, Deputy Director of NVI, who has considerable experience in a variety of vaccine competencies, and our executive officials. Please welcome Dr. Sunet ครับ
Good afternoon, all participants. Welcome to our session in the update of COVID vaccines from global manufacturers. In this session, we have 6 companies are going to present, and we would like to run it in alphabet order. That would start from first with the AstraZeneca, second with Janssen Pharmaceutical, the third with the Moderna, the fourth with Pfizer, the sixth with Sinovac, and the seventh with Sinopharm. I would like to invite our Director of National Vaccine Institute, our moderators, to make an opening of our session. Please, Dr. Nakhon. Dr. Nakhon เปรมศรีค่ะ
Thank you very much, Dr. Sunet, and good afternoon to all the participants. It is our privilege now to invite the international speakers, which are the vaccine developers. I would like to inform all participants this is very special, and it's our privilege to have almost all of the vaccine that now being used in the world, and it will be the very updated information from the directly from the developer, and so you will be get the update based on the dynamic information of each vaccine. So please, this is a very good opportunity of our Thai colleagues as well as our international colleagues who are attending this session. Right now, I would like to start first with the speaker from AstraZeneca, Dr. Bruce Mungall. Dr. Bruce Mungall is an Asia Area Medical Director of AstraZeneca. He is an expert on vaccine and infectious disease. He graduated from University of Queensland as well as a PhD from University of Queensland in 2000. Dr. Bruce used to work in U.S. CDC at Atlanta as a postdoctoral fellow between 2001 and 2002, and then moved to the project leader on pathogenesis of new and emerging zoonotic diseases at the Geelong, Victoria, Australia. and was a visiting assistant professor in pediatrics and infectious disease in Cornell University in New York, USA, and as well as to be the medical communication roles of the MIMS and UBM Medica, which all of the pharmacy and doctor usually use the MIMS and the book for a kind of handbook of the medical doctor, and also used to be the regional and global medical affairs roles in GSK vaccine. Dr. Bruce joined AstraZeneca since June 2021 to the present, and right now he's kindly to join us at the National Vaccine Conference to bring us the most updated information from AstraZeneca. Please, Dr. Bruce.
Sawasdee Krub. Thank you for that kind introduction, and thank you for the invitation to present the available information for the AstraZeneca vaccine, Vaxzevria. Sorry. Small technical glitch. There we go. Okay. Thank you. So in the interest of time, I'll present just a very brief overview of what's happened with a two-dose schedule, but I'll focus very heavily on booster effectiveness. That's the conversation that's going on right now here in Thailand and around the region. So in terms of the two-dose data, we've lost the slide for just a second. Small technical glitch. Okay, thank you. So right from our randomized clinical trials where we licensed the vaccines to start from, and all of the real-world effectiveness data that has been generated over the past year with the use of this vaccine, we've demonstrated quite consistent and quite high effectiveness, particularly against severe COVID outcomes like hospitalization and death across all of the key variants. Now, obviously, with Omicron, the situation has changed, and all of the COVID vaccines are reporting slightly lower effectiveness. But importantly, we're still seeing a considerable boost of antibody responses from 2-dose to 3-dose with Omicron. And importantly, we're starting to see quite good effectiveness numbers coming through from various studies at the moment. Oh, sorry, let me go back one. Just a very brief overview, and I will not talk about all these studies in detail, but there's quite a wealth of evidence being generated for third-dose boosters, both as homologous 3-dose schedules and for heterologous schedules, either with AstraZeneca used first, boosted with something else, or another vaccine boosted as a third dose with AstraZeneca. Let me talk first about the homologous schedule, so 3 doses of AstraZeneca vaccine. And the best available data is coming from the United Kingdom. This is Public Health England data that's available on their weekly reports on their website. And what we're looking at here on the left-hand side is schedules primed with 2 doses of AstraZeneca. On the right-hand side is primed with 2 doses of Pfizer. And so the purple box on the left and the blue box on the right are kind of visual comparisons of 2-dose AstraZeneca versus 2-dose Pfizer. The black dots are effectiveness against Delta. The gray circles are effectiveness against Omicron. And you can see here, visually the estimates that we're seeing for 2-dose vaccines are obviously lower for Omicron than for Delta, but very consistent across these 2 schedules used in the UK. And then in the orange boxes here, both schedules boosted with the Pfizer vaccine. And again, the point estimates that we're seeing for effectiveness against the Omicron variant are very, very consistent across these. Now, very recently, Public Health England has just published a new update where they have homologous 3-dose AstraZeneca effectiveness data. And that's presented for you here from the New England Journal publication last week. And again, you can see here, the orange box is homologous AstraZeneca, 3 doses. So the purple box on the left, if you compare to the blue box on the right, that is 2 doses of AstraZeneca boosted with a 3rd dose of Pfizer. And again, the confidence intervals are a little wider for the AstraZeneca schedule here, and the reason for that is this was a very small group. AstraZeneca was not recommended broadly in the UK as a booster dose, and so the population that received 3 doses were the frailest of the frail. These were people in nursing care facilities who were homebound, etc., and could not go to vaccination centres, so they needed a vaccine that was logistically possible, and so AstraZeneca was provided to those. So that represents a very old, very elderly, frail cohort of about 30,000 people in the UK, which is why the confidence intervals are a bit wider. But obviously the point estimates, again, are quite similar with what we see for 2 doses of AstraZeneca boosted with the 3rd dose of Pfizer. Now, that was for symptomatic infection. If we look at more severe outcomes, this is for hospitalisation in the UK. And again, the left-hand graph is 2 doses primed with AstraZeneca. The right-hand graph is 2 doses primed with Pfizer, and then the yellow box is boosted with a Pfizer vaccine. Again, the numbers very, very consistent across these 2 schedules. So we're seeing quite consistent results in terms of effectiveness for both the heterologous schedule with AstraZeneca on the left or a homologous Pfizer schedule on the right. Now, unfortunately, there's the 3-dose homologous AstraZeneca effectiveness data from the UK around hospitalisation is not yet publicly available. It is coming very quickly, but this is data that's available from the reports at the moment. We will have the 3-dose homologous data very soon, but based on what we're seeing here, I would expect there to be very similar point estimates between the 2 vaccine schedules. Now, what about other schedules? We know that the Coronavac vaccine has been used very, very widely in this part of the world, particularly in Thailand as well. And so if we look at the data for priming schedule with 2 doses of Coronavac and then boosted with AstraZeneca on top, there was a very large study conducted in Chile in South America where the Ministry of Health ran a program to look at effectiveness across their schedule involving about 11 million people in their cohort, roughly 4 million of which were primed with 2 doses of Coronavac, and then 2 million boosted with AstraZeneca, 2 million boosted with Pfizer, and a smaller number boosted homologously with the Coronavac vaccine as well. And they looked at effectiveness across a number of different clinical outcomes. And you can see here, particularly for the AstraZeneca and the Pfizer booster schedules, for hospitalisation, for ICU admission, and for death, very, very consistent effectiveness across these schedules. So reinforcing this consistency of the data that we're seeing in terms of different schedules for these main vaccines in use. Now, the other key schedule, not so much in this part of the world, but in other parts where the Pfizer vaccine or the Moderna vaccine have been used as the priming schedule, there's less data with AstraZeneca boosting, but there is a little bit that has been developed very recently. The COV boost study was run in the UK to look at exactly that in terms of immunogenicity. So they had schedules primed with 2 doses of AstraZeneca or 2 doses of Pfizer, and then they boosted with 7 different vaccines to look at the immune response following that. Here we go. So just to focus on the AstraZeneca data from the COV boost study, this table represents in the top row, we're looking at spike antibody titers. The second row is pseudotype neutralizing antibodies. The third row is live virus neutralizing titers, and we have cellular responses on the bottom row. The first column is 2-dose schedule of AstraZeneca, and this is the standard that we've had for our previous clinical trial results and all of our effectiveness data. And I just want to reiterate the numbers we see here have been associated with between 72 and 92% effectiveness against severe disease caused by the various variants of concern. If we look at the 3-dose homologous schedule of AstraZeneca in the second column here, you can see that the numbers for each of these endpoints have increased substantially. And so the effectiveness that we would expect from those values is clearly going to be higher than the 2-dose. But importantly, on the right-hand side here, for schedules that were primed with 2 doses of Pfizer and then boosted with AstraZeneca, you can see, again, a significant increase in the value of all of these titers, 5 to 6 times increase. So even though we don't have that effectiveness data from real-world studies looking at that specific schedule at the moment, based on the immune responses that we're seeing from the COV boost trial, we would expect effectiveness at least as good as we've seen with the 2-dose schedule with those lower titers. Now, just a quick word on safety. It's very important, I think, given the billions, literally billions of doses that have been rolled out for these vaccines to continue to monitor safety very, very carefully. And importantly, with third doses, I think people are a little bit nervous, I guess, about repeated doses of vaccine. So I just want to reassure you that in all the data that we've seen so far, a third dose of AstraZeneca, whether it be a homologous dose on top of 2 other AstraZeneca or heterologous on top of another vaccine, there have been no additional safety concerns identified. It's quite well tolerated. We know with heterologous schedules there is slightly higher local reactogenicity, but nothing that's concerning. These are quite mild and resolve in most patients very quickly. So no real concerns with an additional dose of the vaccine. I just want to touch briefly on the rare safety events. We know that the viral vector vaccines, both AstraZeneca and the Janssen vaccine, did have some associations with blood clotting events, particularly in Europe early on. There has been some very large studies conducted just in the last couple of months to evaluate the real impact of those and whether those events are actually as common as was initially feared. This was a study from the UK, published just very recently, looking at 46 million adults in the UK. And they measured the hazard ratios for venous thrombotic events and for arterial thrombotic events here. And because the 2 main schedules in the UK were AstraZeneca and Pfizer, they've measured this for 2 doses of both of those vaccines here. And you can see very clearly, I think, from this graphic that the hazard ratios for both vaccines were very, very similar for all of these key events. They also looked at thrombocytopenic events, because that's one of the associations as well that was drawn initially. And you can see here for the AstraZeneca vaccine, there is a very slightly elevated risk across these, depending on the stratification for age or for sex. But again, very minimal in terms of increased adverse events. So the safety profile continues to remain very positive, I think, for this vaccine. And given that we have already distributed in the ballpark of 2.7 billion doses globally, it remains a very big contributor to what we're seeing. Now, the last point I want to touch on very, very quickly is around the conversation that we continue to have around antibody responses for these vaccines and what that may or may not infer in terms of protection. Just a quick snapshot. This is data from the UK. On the left-hand graph, you're looking at antibody titers against the spike protein for the Pfizer vaccine in blue and for AstraZeneca in the pink bars you can see here. And we've seen this data many times. There are significant differences in terms of the antibody responses that these vaccines do produce. But importantly, if you look at the graph on the right-hand side, this is effectiveness data against hospitalizations due to Delta in the UK as well. And you can see after 2 doses of these vaccines, the point estimates are very, very similar with no difference at all. We see a very similar picture in Thailand, looking at booster schedules. So the graph on the left is looking at subjects primed with 2 doses of Coronavac in the light blue dots at the bottom. In the blue box at the top, from the left to the right, they were primed with AstraZeneca, a half-dose Pfizer or a full-dose Pfizer. And again, you can see from the antibody responses, there are significant differences here that we see in terms of the immune response. But looking at the effectiveness data on the right, also generated from regional Thai studies here, for the equivalent schedule highlighted in the blue box, 2 doses of Coronavac boosted with either AstraZeneca or Pfizer, we see very, very similar point estimates for effectiveness, both from the Bangkok and the Chiang Mai studies. So I think the conversation that we continue to have around measuring antibodies, because we think that gives us a measure of the protection, we need to qualify that a bit with what we're seeing in terms of real world effectiveness. And the data we've seen so far has indicated that the key vaccines, certainly the AstraZeneca vaccine and the mRNA vaccines, provide very, very good and very similar protection, particularly against severe COVID outcomes. Thank you for your time.
Thank you very much, Dr. Bruce, for the presentation. I think that Dr. Bruce has touched to several points that are very important, that including not only the immunogenicity data, not only the safety overall data, but also the real-world effectiveness, particularly in the UK studies. So I mean that is very, very important because Thailand has been used AstraZeneca vaccine a lot. So particularly that informed that the booster with the viral vector still confer to the effectiveness of the more than 80% of reduction of hospitalization. That is, I think that this is the key taking point. And I think that there would be some other question that may come from the audience, but I would let that to be at the end of our session. Let me introduce for the second speakers for the Janssen Pharmaceutical, Dr. Maria Carmen, for the second speaker of the company that have the viral vector vaccine as well. Dr. Carmen has a long experience in the vaccine and in the pediatric infectious disease in the Philippines. She has worked in the vaccine field for more than 10 years, has even worked in the pharmaceutical, in the clinical development for vaccine for the pediatric vaccine, including not only in the country, but in the regional level and in the global level. At the moment, Dr. Carmen is the Asia Pacific Regional Medical Affairs Director in infectious disease and vaccine. Please join the presentation of Dr. Carmen.
Good afternoon. I am Carmen Nievera, representing Janssen Asia Pacific Medical Affairs. We thank the NBI for the kind invitation to present our real-world evidence on Janssen COVID vaccine with a particular focus on variants of concern. Before I begin, these are my disclaimers. The vaccine I am discussing has not been approved for commercial use by regulatory authorities in Thailand. Kindly refer to your local prescribing information and national recommendations. This is the outline of my presentation. After a brief background, I will be presenting real-world evidence on effectiveness with focus on variants of concern. Thereafter, I will discuss safety before I end with our conclusions. Development of our vaccine began in January 2020, immediately after the virus sequence was available. The vaccine underwent a comprehensive clinical development strategy, initially evaluating safety and efficacy of a single dose for epidemic or pandemic response globally. Single-dose efficacy was 75% against severe disease and 85% against COVID-related deaths, and has persisted for up to 6 months. Antibody titers rise over time and persist, and cell-mediated immunity is especially strong with CD4 and CD8 T-cell responses that are likewise durable. A booster further increases protection against COVID. And so here are some of our Janssen-sponsored studies determining the vaccine performance as homologous or heterologous boost in terms of safety, immune response, and efficacy. And here are non-Janssen-sponsored studies done in collaboration with external investigators. We are happy to be collaborating with the NBI and IVI in one such study which is ongoing in Thailand. Current data show that a booster dose has acceptable safety profile similar to single dose. Further, efficacy against symptomatic disease increased, and there was complete protection against severe disease. The booster dose increased antibodies against all variants tested, including Delta and Omicron. Given this data, the US FDA granted emergency use authorization for the vaccine as homologous and heterologous booster. So we now move on to real-world evidence on effectiveness during periods of different circulating variants of concern. Here is a summary of US data after a single dose of the vaccine. To date, there are at least 11 studies from different investigators, and I present only data on Janssen COVID vaccine in these slides. The blue shading indicates a high Delta time period. Vaccine effectiveness estimates differ from study to study because of the different data sources and methodologies. Before Delta predominance, vaccine effectiveness ranged from 68 to 77% against infection and higher against hospitalization. Studies done later during high Delta circulation showed vaccine effectiveness of 60 to 88% against infection and 71 to 81% against more severe outcomes like hospitalization. Lower vaccine effectiveness was seen among immunocompromised. CON and others studied vaccine effectiveness among veterans. Here, analysis from Feb to August. And here, continuing to October, findings showed decline in effectiveness versus infections. The same study, though, found effectiveness against death at 73% and 52% in those 65 years and older. Now, let me highlight some interesting findings from a few of these studies. First is a retrospective medical claims analysis of a cohort of 390,000 adults who received a single dose between March and July in 2021. Vaccine effectiveness against Delta variant was assessed in a cohort of around 29,000 from 4 US states with early and high prevalence of Delta. Results showed effectiveness at 79% against infection and 81% against COVID-related hospitalizations. Looking at effectiveness over time, on the left-hand side, you see month-over-month vaccine effectiveness for COVID-19 infections as well as hospitalizations. Vaccine effectiveness was consistently stable month-over-month across the entire study period, including in the pre-Delta time frame as well as during Delta predominance, seen in the green box for the months of June, July, and August. On the right-hand side, the Kaplan-Meier curves for the time to event analysis for COVID infections demonstrate a stable vaccine effectiveness at least 152 days post-vaccination, even amid high Delta dominance. Another study looked at medical claims data from January through to September 2021 in more than 17 million adults. This case-controlled study looked at odds ratios for developing breakthrough infection, hospitalization, or ICU admission from 2 to 6 months following vaccination relative to the vaccine effectiveness in the first month after vaccination. For those vaccinated with the AD26 vaccine, there was no evidence of waning protection against breakthrough infection in the first 3 months following vaccination, and some modest waning protection was seen in months 4 and 5. Further, for COVID-related hospitalization and ICU admissions, there was no evidence of waning protection. Thus, the vaccine showed strong and durable protection against breakthrough infections, hospitalizations, and ICU admissions throughout the study. In summary, all 11 studies showed real-world effectiveness of the vaccine against COVID infection and important outcomes such as hospitalization and death, and with signs of durable protection even during Delta predominance. Now we look at data from more resourced, limited setting like South Africa. In an open-label, single-arm phase 3 study, we monitored the safety and effectiveness of one dose against severe COVID, hospitalizations, and deaths in 500,000 healthcare workers as compared to unvaccinated healthcare workers in South Africa. This is part of Sisonke studies, which assessed real-world effectiveness of both single-dose and homologous boost of the vaccine. We assessed effectiveness of the single dose during Beta and Delta wave, and then we assessed effectiveness of 2 doses during Omicron wave. For the single dose, overall VE was 83% to prevent COVID deaths, 75% to prevent hospital admissions requiring critical care, and 67% to prevent COVID-related hospitalization. The VEs were consistent in older healthcare workers, those with comorbidities, and throughout the Beta and Delta dominant phases of the study. In the next slides, we move to the 2-dose effectiveness, also from Sisonke study. But just to note that data from other studies seen here support that homologous and heterologous boost combinations do increase humoral and cellular immunity to Omicron and can be considered as a strategy to mitigate Omicron risk. So we move to Sisonke 2 study. where here a homologous booster was given 6 to 9 months after the initial vaccination, and we evaluated vaccine effectiveness in preventing hospital admissions. The study enrolled 220,000 healthcare workers from November to December before the onset of Omicron wave, affording us opportunity to evaluate VE against Omicron. After booster, VE against hospitalization was 63% during the first 2 weeks and increased over time to 85% 1 to 2 months post-boost. These findings reassure that a homologous boost is protective against hospital admissions, including in Gauteng, the epicenter during their Omicron surge. This provides first evidence of the effectiveness of a homologous boost during Omicron circulation, with effectiveness increasing over time. So let's move on to safety. The Sisonke study also provided real-world evidence on safety. Of almost 500,000 participants, only 2.2% reported AEs, and 1.4% of these were SAEs. Most were reactogenicity events, most commonly headaches and body aches, and most occurred on the day of vaccination and were reduced by day 2. Serious AEs and AESIs occurred lower than expected population rates. Out of the AEs examined, TTS and GBS occurred at rates greater than the baseline comparison population, with observed versus expected ratio of 2.4 and 5.1 respectively. For the other AEs, the observed to expected ratio was less than 1. 2 TTS cases were reported. Both were in women with risk factors, and both participants recovered. The authors concluded that the single dose is safe and well tolerated in adults, with few serious adverse events, which were successfully managed with prompt identification in South Africa. A robust pharmacovigilance system is important to enable continued assessment of the risk-benefit profiles of COVID vaccines. Since the EUA, the following events have been added to the US or EU labels based primarily on post-authorization safety reports: coagulation disorders such as TTS, venous thromboembolism and ITP, GBS, and capillary leak syndrome. Data presented at the US VRBPAC meeting last October showed the following post-authorization reports of potential TTS and GBS worldwide. Following Janssen's review of available information and based on CDC case definitions, there was a reported rate of 2.1 TTS cases per million doses and 7.5 GBS cases per million doses. Regarding safety of a booster dose, we find more real-world data from the US. The CDC reviewed safety data of boosters from September to February 2022, with data coming from VSafe and Bayers, with 82.6 million adults in the US having received a booster dose. Focusing only on Janssen vaccines, findings for booster vaccination are like those described in clinical trials, in that less reactogenicity is seen with homologous boosting than with heterologous boosting. In conclusion, clinical trials have demonstrated durable protection of the single-dose vaccine with increased efficacy with a booster, including against circulating VOCs. Real-world evidence from different settings showed effectiveness against COVID, especially against severe outcomes such as hospitalization and death. Data also suggests durable protection over time. Preliminary safety findings are like those in clinical trials with no increase in AEs after booster. TTS is a very rare but potentially life-threatening adverse reaction associated with the vaccine. More data are being generated, but current data support a positive benefit-risk profile during the pandemic setting. Thank you very much for all your kind attention.
So thank you very much, Dr. Maria Carmen. We have got a very informative exchanging information from the real world data of using Janssen vaccine. And we have seen that the vaccine still be benefit with the homologous booster or the heterologous booster, and the safety is to be outweigh the risk. So the benefits to be outweigh the risk of even the TTS or the Guillain-Barré syndrome, and the effectiveness of preventing hospitalization and severity of Janssen vaccine by homologous booster or the 2 doses, or even the heterologous boost on the other vaccines to be very satisfied. So this is another one of the I would say viral vector platform that have shown the effectiveness that we can share to the participant and to the world. Thank you very much for your kind providing the information. I would like to move to the next speaker that we've got the privilege from Dr. Christopher Clarke, the Director of Scientific Leadership of Moderna Therapeutic in Asia Pacific. And Dr. Clarke finished the PhD in the School of Pathology in Imperial College of School of Medicine in London, UK. And as a Scientific Director of Moderna Asia Pacific region, Dr. Clarke is supporting the role in the rollout of the mRNA 1273 or Moderna vaccine and development of Moderna mRNA vaccine pipeline. Dr. Clarke brings more than 13 years experience in vaccines industry, during which time he has participated in the development project leading up to launch and implementation of a number of innovative products, most prominently influenza, pneumococcal, herpes zoster vaccine into the national immunization programs in Australia and across the world. Prior to moving into industry, Dr. Clarke was an academic researching immunological and molecular mechanisms linking innate and adaptive immunity, particularly in context of viral infection and cancer. Over his career, he has published research in more than 30 papers relating to infection, immunity, and vaccines. So right now, we would like to invite Dr. Christopher Clarke, please.
So thank you very much for that kind introduction. In this session on real-world evidence, I was asked to focus on the data relating to the booster doses of Moderna's SARS-CoV-2 vaccine mRNA-1273 in the context of the current challenge where Omicron is the dominant circulating variant. So I'm going to highlight some points about safety and efficacy in this scenario that might be of interest. And if I have one key message, it's that use of a booster dose of COVID vaccines is important, particularly for the Omicron strain. So as a brief introduction to the mRNA-1273 vaccine, it contains an mRNA encoding the SARS-CoV-2 spike protein from the ancestral strain of the virus. And it's formulated into a liquid nanoparticle, which allows delivery of this mRNA into the cytoplasm of antigen-presenting cells. And this allows for production of the spike protein in the host cell, which acts as the antigen to generate a protective immune response to the virus. So the product is based on an RNA platform that's been developed over the last 10 years. And this is really what's allowed the very rapid progress of the vaccine development at the start of the pandemic. Now, before we get into the real world evidence part of the talk, I just want to remind you briefly of our large phase 3 COVE study, which was the basis for emergency use authorities across the world. It was a randomized observer blind trial that enrolled over 30,000 adults in the US who received either a vaccine or the placebo. The 100 microgram dose of 1273 was generally well tolerated. Most common side effects were pain at injection sites, fatigue, headache, myalgia, and arthralgia, more frequent after the second dose than the first. And at the interim analysis of efficacy, which was what was the emergency use authorization was based on, we saw efficacy of 94% against symptomatic infections. Now, even though that study is actually still going in terms of follow-up, it does feel a bit like ancient history because so much has happened over the last 15 months. The first emergency use authorization was in the US in December 2020, and the vaccine was finally approved in the US in January this year for adults. The 100 microgram dose has since been authorized for use in adults and adolescents in many other countries worldwide, including Europe, Japan, Australia, and many other countries of the Asia-Pacific region. The 50 microgram dose is approved for use as a booster for adults in many countries. And this dose has also been approved for the primary course in school-aged children in Australia and received CHMP positive opinion in Europe for that same population. So lots has been happening, adding up to more than 807 million doses distributed in 2021 and more coming in 2022. So in line with what was seen in our randomized controlled trials, the initial real-world evidence did show that COVID vaccines were very effective against the ancestral strain and against the early variants, particularly against severe disease. And you've seen a lot of examples of that in the first two presentations. But in the second half of 2021, the emergence of the more transmissible Delta and Omicron variants really did pose a new challenge. And that's really the reason why we started introducing booster doses. So the data I'm showing you on this slide are taken from an additional analysis of a collaborative study between Moderna and the Kaiser Permanente healthcare organization in Southern California. And it was conducted over a 3-week period in December, just as the Omicron variant was taking over from the Delta variant in the US. So you can see overall effectiveness against symptomatic infection was about 64% for Delta and around 14% Omicron. But this was declining over time. From in the earliest time points, we're seeing 80% and 44%. By the time you get out past here to 9 months post-vaccination, we're really looking at much lower numbers, and particularly for Omicron, 6% of vaccine effectiveness against Omicron for these patients who are vaccinated more than 270 days previously. And similar, it was not really unexpected to see this lower vaccine effectiveness against Omicron. It has many more mutations than the other variants, including 15 in the receptor binding domain alone, and that's an important antigenic site. So based on this and similar observations with other vaccines, it wasn't really a big surprise that we saw those additional waves of infection towards the end of 2021 and into 2022, including in countries where there was very high coverage of primary vaccination schedules. So considering the limited protection that's being offered by the primary series against Omicron, particularly over time, why might using a booster containing the same antigen have any benefit? This graph is showing you neutralizing antibodies measured by a pseudovirus assay against the ancestral strain in black and against the Omicron strain in red. And it's for a subset of individuals who are followed from before the primary series until 6 months post-booster. So here we're looking after the primary series, and you can see the levels of antibody are much lower for the Omicron strain than they are against the ancestral strain. And they do, for both strains, they do decline over time. But what was encouraging in this study was that the 50 microgram booster does restore both the neutralizing activity of the ancestral strain, but now we're seeing neutralizing antibody levels that are actually very much approaching what we saw from the ancestral strain after the primary series. So this gives us some encouragement that potentially we would see effectiveness of the booster dose against Omicron. But these, of course, are immunological data, and it's important that we study protection. So there is effectiveness data emerging in different parts of the world, and I'm not going to go through it all. I'm just going to show you a couple of examples. And this data I'm showing you here does come from the same study I showed you previously, conducted at Kaiser Permanente in Southern California. And if you remember, I showed you that 270 days after a primary series, the vaccine effectiveness had declined against Omicron to around 5%. And what we're seeing now is that the vaccine effectiveness initially up to 72% and was 70% overall over the whole study period. And these are vaccine effectiveness estimates against infection, and the numbers are actually even higher when we look at hospitalizations and severe disease. So those data from Kaiser Permanente do refer to groups of patients who'd received a 1273 booster after a 1273 primary schedule. But in many countries now, people are now receiving different vaccine as a booster to the one they received as a primary course. So you'll have seen similar graphs to these in a previous presentation. They come from the Health Security Agency in the UK, and I'm showing you vaccine effectiveness against hospitalizations over time in England, where the vast majority of people received either a Pfizer or an AstraZeneca vaccine for their primary courses. And similar to what I showed you in Kaiser Permanente, both vaccines initially offered some protection against Omicron after the primary schedule, but this appears to be much lower. So the Omicron is here showing you at the bottom in the open circles. And this is on the left-hand panel, we're looking at people who are primed with the AstraZeneca vaccine. And on the right-hand side, we're looking at people who are primed with the Pfizer vaccine. Yes, the Omicron in open circles and Delta strain in black squares. So we're seeing some protection against hospitalization, but it does decline over time. And this is last time point here in both cases is after 6 months. So similar to what we'd seen in the Kaiser Permanente. And in England, as is already mentioned, for the booster dose, mainly RNA vaccines have been used in the booster campaign so far. There is some AstraZeneca, but much more limited. So what we see when we look at those who received either of the RNA vaccines, either the Pfizer vaccine or the 1273 vaccine, is after we give the booster dose, we see a sharp increase in the vaccine effectiveness against hospitalizations for Omicron and for Delta above what was seen after the primary course. And this is again on the left-hand panel for AstraZeneca primed people, on the right-hand side for Pfizer primed people, but the effectiveness data is looking very similar regardless of which priming that's occurred. So to assess what this meant for the healthcare system, the Health Security Agency also modelled hospitalisation cases prevented in England by their booster campaign. The blue line shows the observed cases and the orange line shows the modelled expected cases. And the difference between the two lines represents the reduction in hospitalisations. So this adds up to a reduction of around 130,000 hospitalisations in the population aged 25 and over in England in this short Omicron dominant period alone between December and February. So this model really gives a concrete demonstration of how the booster programme, primarily with RNA vaccines in England, was associated with substantial reductions in healthcare use. So safety of the booster dose is also an important consideration. And the data we had from clinical trials had shown that adverse reactions in response to a 50 microgram booster were similar in nature, but perhaps slightly less common than seen after the second dose of the primary course. And this has been reaffirmed in what we've seen in the post-marketing setting. On this slide, I'm showing you some post-marketing data from the US, which is a country where most doses of 1273 have been distributed. On the left-hand panel is showing you rates of adverse reactions by dose within 7 days of vaccination, and these were collected in a patient-reported system using a SMS. Overall, adverse reactions are quite common, but only very few of these reactions actually require medical care. And what we're seeing is that rates after the third dose, the booster dose, do appear slightly higher than after dose 1, but they are in fact lower than after dose 2. So this review also included data from the VAERS database in the US, looking at the frequency of rare adverse event myocarditis. After the booster dose, this is mainly affecting young males, peak incidence aged 18 to 24 at a rate of 8.7 per million. And it's close to background rates in the other age groups. Important for the discussion is that the rates after the booster dose were much lower than the 56 cases per million reported after the dose 2. which was already a very low incidence. We also have data on the incidence of myocarditis and pericarditis by dose coming available from Japan, which is another country that used large numbers of doses of 1273. And as in other data sets, the overall incidence of myocarditis and pericarditis is very low. It's higher after the second dose than after the first dose. But as the booster dose program started much later in Japan than in the US, the number of exposures is quite low. And as of the 23rd of January, there'd actually been no reports of myocarditis and pericarditis following the booster dose of 1273 in Japan. And this is, but the rates are somewhat consistent with what would have been expected based on what we saw in the US. So assessments of very rare adverse events after vaccinations do need to consider many factors which contribute to benefit risk evaluation. And the first point is to say that cases of myocarditis of the vaccination do appear to be mild and generally resolved with conservative management, usually with non-steroidal, with NSAIDs. And there's no evidence that these have long-term effects as yet, though I think more time and more study is needed. We also need to consider that episodes of myocarditis have been associated with COVID infections. In fact, they occur at much higher rates after COVID infection than they do after vaccination. And lastly, it's important to weigh any risk of adverse events against the substantial burden of disease that's being prevented by vaccination. So it's a very relevant point that the Omicron wave has been characterized by large numbers of infections, hospitalizations, and deaths. So I think my time is almost up, so I'm just going to give a brief summary. So I've talked about the issue of waning effectiveness and how that's particularly important with respect to Omicron, and actually looking at effectiveness data that's relating to your actual strain of interest rather than what's happened in the past is really important. So, and what's more, we also need to look at the effectiveness that we have on our current vaccines in the context of the Omicron wave as opposed to previous strains. I've also talked about the safety of booster doses, and what we've seen here is the pattern of events observed appears similar to what's been seen for primary courses. Events after dose 3 do appear to be less frequent than after dose 2. And the last thing I'll leave you with is about the future. It is widely considered that other variants will follow Omicron, and potentially we might have to think about additional doses moving forward. And it's really time to start planning about how to address those challenges right now so we're ready when they come. So thanks very much for listening, and I think we'll be taking questions at the end.
Thank you very much, Dr. Chris Clarke. I think that Dr. Clarke has mentioned the key messages from his presentation, particularly the mRNA Moderna vaccine booster in this Omicron period can save a lot of hospitalization and could help for the public health during our Omicron outbreak. The second point Dr. Clarke has mentioned about the booster of 50 microgram has worked well in several, in many countries that has been used this vaccine, particularly Australia, Japan that even use this vaccine for adolescents as well. Dr. Clarke has mentioned a lot on the immunogenicity, particularly neutralization against the Omicron, and it worked well and showing into the real world effectiveness in the UK studies that use the Moderna vaccine boost on the primary vaccination, both Chadox or the Pfizer vaccine. He has even talked about the confirmation and to ensure that the safety profile of the booster of the mRNA vaccine is similar to the first two doses of the primary vaccination or even lower. I think that there would be other questions that would come later and we would pick up to answer at the end. Let me introduce for the next speakers, Dr. Egemen Ozbilgili from Pfizer. Dr. Egemen, he is the Senior Director of Vaccine Regional Medical Lead. He has a long experience, more than 10 years in the Pfizer Turkey for working on the burden of pneumococcal infection in Turkish children and led the national pneumococcal surveillance studies with numbers of pediatricians and microbiologists in Turkey. He has worked with the responsible to work with the National Immunization Program decision in Turkey and expand his work on the Pfizer-Israel vaccine during 2015 to 2018. And then he moved to work with the Pfizer-BioNTech COVID vaccine mRNA vaccine phase 3 clinical trial program in Turkey. And then he moved to Asia to be the role of the regional medical lead for Pfizer vaccine. So please, Dr. Egemen, for the presentation.
Thank you very much for this nice introduction. Good afternoon, good morning, everyone. So most of the data are common with all the counterparts from the other companies. So I will present some similar data with some different perspectives maybe, but I'm really happy that we have some common messages to all the scientific community. So I prepared my presentation starting with safety. So it's a good link after Dr. Chris. He also mentioned a lot at the end with the safety. So I will maybe summarize or say the same data set. So here's the data. Let me start it from United States, over 18 years of age. Safety of booster data of BNT1622 mRNA vaccine in almost 16 million doses. So on the left hand, you see the 16.2 million doses administered Pfizer vaccine and 26 million almost all vaccines administered in the time period of September to November last year. So there were 600 serious adverse events and 473 adverse events in both groups with some similar death results. So all are wondering about the myocarditis report. So here is the result for myocarditis in this age group. There were 12 cases met the CDC case definition. So 8 with Pfizer and 4 with Moderna vaccine with almost 46 years median age. So 6 of these 8 recovered from symptoms at the time of the reporting. I will also go into the details for myocarditis in the upcoming slides. So booster profile is consistent with the reactogenicity following with the primary series, and you see on the local and systemic reactions chart on the right. And this is the data again from United States in the adolescent age group with 2 doses scheduled. So there were 14 deaths and almost 849 serious adverse events reported again through the US vaccine adverse event reporting system. So most report of serious events are consistent with the diagnosis of myocarditis. So these findings are consistent with our clinical trial safety data, except myocarditis, which is a serious adverse event detected in the post-authorization monitoring. And now none of the reports of death were determined to be the results of myocarditis. I'm talking about 14 cases here. And here is the rated result for children age group 5 to 11 years of age with 2 doses. So we don't have still 3 doses in this age group, you know. So the data is from November to December last year. The findings, I'm not going into details, but consistent with our clinical trial safety data in the same age group. Administration errors without adverse events is the most common report in this age group, most likely due to the first receipt of lower doses, which is 10 microgram when you compare to the 30 microgram in the adult age group. There were 2 deaths observed in that report. One is 5 and the other one was 6 years of age, both who had some underlying medical conditions, medical histories, and very fragile health before vaccination. So none of the data suggested a causal association between that and vaccination. And this is the data from Israel, over 16 years old. This is a nationwide mass vaccination report, you may recall from the Israel data. So on the left, you are seeing the risk ratios for some adverse events after vaccination or COVID infection, like acute kidney injury, like deep vein thrombosis or myocardial infarction. So it's clearly seen that infection itself is more risky when you compare to vaccination. On the right-hand side, when we take into account myocarditis, the blue bar is risk difference per 100,000 persons who received Pfizer mRNA COVID-19 vaccine, and on the orange one, you're seeing the risk difference for the same population infected with COVID. So it's clearly seen that infection is much more risky than vaccination itself, even for myocarditis. And here is the data from UK. This is the population-based prospective study using the UK National Immunization Database for COVID vaccination. So here are the results on the left-hand side. For each of the vaccines, already all the counterparts from different companies nicely presented. So for each of the different vaccines, roughly, it's like 1 to 6 or 10 per million in terms of myocarditis events per 1 million. However, when we assess the infection itself here, it's again, like in Israel, it's much more higher with an infection, both for myocarditis, pericarditis, or cardiac arrhythmia. So we can easily say that regarding the risk-benefit ratio, vaccination itself is much more beneficial when you compare to COVID infection for myocarditis. And let me go through some effectiveness data for Omicron. So this is the data with the third doses of BNT162b2 with the sera of human sera of third dose. On the left-hand side, you're seeing the pseudovirus neutralization results for which wild type and for Omicron. As we know, after the second dose, there is not enough neutralization antibody against Omicron. However, 1 month after the third dose, post-third dose, the results are similar with the results after 2 doses against wild type. And these are similar with the live Omicron virus. So there were 22-fold drop in Omicron neutralization. And after the booster dose, 23-fold increase against Omicron with sera in pseudovirus. Again, some similar results for the live virus. So now we will see the reflection in real life settings. This is the early data already presented recently from UK. So with 2 doses against hospitalization, vaccine effectiveness is 72%. And with time, as expected, there's a waning immunity, which comes down to 52%. And after 2 doses, 3 doses, and after 2 weeks, it again increased to 88%. So we know that there is a waning of immunity after 2 doses against Omicron. So this is why we so strongly work on the third doses of data for all these vaccines, and strongly recommend the importance of booster dose in the Omicron era. And it's another data from United States reflecting the time period between August of last year and January of this year with any mRNA vaccines. So as we know, the effectiveness was high even with 2 doses in the Delta period. And with Omicron, we know that the hospitalization or emergency department vaccine efficacy against emergency department use of hospitalization is still high with the 3rd doses. This is again very similar, very in line with the data we saw from UK. And this is the data from Kaiser Permanente. It is showing the vaccine effectiveness in the period of Delta and Omicron separately. The time period was December 1st of last year to January 11. So after 2 doses, emergency room visits without hospitalization, the vaccine effectiveness was much higher for Delta and as we expected, lower for Omicron. And this is decreasing with the time period. When it's almost 5 months, the vaccine effectiveness is 38%. So then after the 3rd booster dose, it again increased to 78%. And again, with the time, decreasing almost to 50% levels. But it is still high against hospitalization, which is the most critical parameter that we keep in mind, that we expect from vaccine. So after the 3 doses, the vaccine effectiveness is 89% with a median follow-up, 4.1 months. This is a very recent data from the United States. This is a prospective observational study comparing 2 groups. Delta period is 4th of July to 25th of December last year. And after 25th of December, they called like Omicron period. And the data is till 14th of January of this year. This is just published recently last week, reflecting the 12 state data from US. So as we've seen in the previous data, vaccine effectiveness with 2 doses against Delta was 85%, and it is similar, again, even higher with 3 doses. And with Omicron, it's almost 65% for 2 doses, and it increased to 86% after the boosted 3rd dose. As we now understand from our clinical practice in all over the world, the in-hospital mortality was 7.6% for Alpha, 12.2% for Delta, and 7.1% for Omicron. So Omicron is a little bit more milder when you compare to Delta. However, the numbers are huge in all over the world because of its fast transmission. And the last data from UK, again, presented recently, but I just want to highlight some results for Pfizer vaccine. So the period is almost beginning of December last year and January 12th. It's almost 1.5 month period in UK, in England. And these are the vaccine effectiveness results for Pfizer vaccine. I want to focus on 2 dose and booster dose results. In line with United States, the vaccine effectiveness was 65% and waning timely. Almost after 25 weeks, it's almost 8%, which is very, very low. And after booster, again, similar with US data, it increases to 67% and still remaining same in the first month, but then starts to decrease to 50% levels after 2 months. So these are the data, real world evidence for Omicron. And lastly, I want to cover, which is another interesting topic for Pfizer vaccine, I guess, especially the children and adolescent age group. I want to focus mostly for the children part. So let me remind you the efficacy results of BNT1622 in children 5 to 11 years of age with 2 doses. As we remember, this was the time for Delta. There was no Omicron and we were running that efficacy trial. And the efficacy result was 90% in this age group, 2 doses. And now we have one of the first recent results from CDC, just published last week. I want to focus you 2 groups. One, the most interested part, we are waiting the first results for 5 to 11 years of age during the Omicron period. As we know, we are just administrating our vaccine 2 doses now in this age group, so there is no 3rd booster dose yet. So this is the data I'm underlining with just 2 doses in 5 to 11 years of age. And the vaccine effectiveness is 50%, 51% in this age group in the early periods after 2 weeks or 2 months. And with the older group, again Omicron, we have booster. We have 3rd doses for 16 to 17 years of age in US. So it's recommended there. So after the 3rd dose, the vaccine effectiveness is 81%, which is in line with the expected data. So this is my last slide. Just as a conclusion, we have very strong and good vaccines from many different companies, and now we have very, very detailed effectiveness and safety data for each of these vaccines. And now we are all working on the importance of booster in many countries. So I think this is a very good opportunity and very good time to discuss maybe in the Q&A session how we can leverage, how we can strengthen the vaccination, especially in the Asia. Thank you so much.
So thank you very much, Dr. Egemen. This is very, very updated information that we've got from the vaccine developer. So right now we have 4 vaccine developer, and I can grab the message that we have had so far from 4 developers, 2 viral vector vaccine and 2 mRNA vaccines, that the 2-dose regimen is not enough for now in fighting against Omicron strain. We do need the 3rd dose booster in every vaccine platform. Or let's say for the Janssen vaccine, it may need a booster dose from the original regimen of the single dose. So in short, that we do need the booster dose of vaccine in every person. And we observe the declining trend by time of the effectiveness in preventing both of infection and hospitalization, meaning that we do need our strengthen and tighten of the other prevention measure as well. And for the safety, we have seen large number of safety profile of vaccines that can provide us the information between risks and benefit of the vaccine. So right now we will move to the other 2 conventional platform of vaccine. So I would like to introduce the next speaker, Mr.Weining Meng. He is the Vice President of Sinovac Life Science. He has an expertise in biopharmaceutical industry, and he obtained his master's degree from University of Queensland. Mr. Meng has more than 18 years experience on biopharmaceutical industry. He joined Sinovac since 2007 and has worked as a quality assurance manager, international regulatory affairs manager, and director in Sinovac company. Under his direction, Sinovac vaccine has successfully been registered and exported into more than 15 countries. In 2017, Sinovac hepatitis A vaccine has been pre-qualified by WHO, which is the first Chinese hepatitis A vaccine pre-qualified by WHO. Currently, Mr. Meng is Vice President of Sinovac Life Science, and he is leading team to conduct a clinical overview of COVID-19 vaccine, namely CoronaVac. And currently, CoronaVac has been approved for use in more than 50 countries. So please join us to welcome Mr. Weining Meng from Sinovac Life Science, please.
Good afternoon, friend from Thailand. This is Weining Meng speaking. I'm from Sinovac Life Science. I'm the Vice President of Sinovac and taking charge of the international regulatory affairs and some business development. So today I would like to give you some update about our COVID vaccine CoronaVac, the safety and effectiveness in the real world. Firstly, let's see the total picture of the usefulness of CoronaVac globally. So until end of January, the CoronaVac has been approved in more than 60 countries and regions, including the WHO EUL approval. And regarding the doses distributed in the world, more than 2.7 billion of CoronaVac has been supplied globally, and including more than 1 to 6 billion in China mainland and Hong Kong, and also around 1.1 billion distributed outside of China. And among those huge doses, actually 2.3 billion of those has been used in the people's arm and play a quite important role for the population against the COVID. So total those numbers is around 25% totally COVID vaccine dose used in the world. So based on those large vaccinations of CoronaVac through our pharmacovigilant system, also by collaboration with other stakeholders in different countries, we collect the safety profile about this vaccine. We can see CoronaVac, since CoronaVac is the time test technology, which is the inactivated viral vaccine, and so the safety profile of CoronaVac is similar with others similar technology product. The safety is quite good. You can see they are comparing with others COVID vaccines with the other technology platform. Obviously, this is the biggest advantage of CoronaVac. So the total, the EFI incident in general is much lower, and also even comparing with other inactivated vaccines, such as inactivated hepatitis A, seasonal influenza, hepatitis B vaccines, the serious EFI incident rate is quite similar. And so through those data, we can see the safety of this vaccine is reliable, and obviously if the vaccine are used in the whole population, including elderly, adult, even pediatric, and the safety profile sometimes it's quite important for the decision makers to give some special populations such as the chronic disease patient, elderly, and pediatric group. So next, I would like to share some data about the effectiveness from the real-world situation. First, let's see the data from Brazil. This is the quite in the middle of last year actually, when CoronaVac was approved by Brazil Anvisa, and so our collaborator, Butantan Institute, conduct large real-world studies in a small town called Serena. That's Serena city located in São Paulo, Brazil, and it's a total about 45,000 population, and among them is about 28,000 adults. At that time, only adult, the vaccine only approved for use of adults. These studies are mainly conducted in adult population. So this city also quite special because about 25% of the population is working outside of the city. So that means that those people actually travel around every day between the city and some towns outside of the city. And we are told that finally we in these studies we design about 30,000 subjects could be enrolled in these studies, and vaccination schedule is 2 doses with 0-20 days, which is widely used in the world. And then the vaccination was given on a different period, including you can see step 1, step 2, step 3, step 4 from different area of the city. And then after that, all the subject entered in the monitoring period and checking the efficacy, effectiveness of this vaccine in this population. Finally, more than 27,000 subjects enrolled and get the vaccines. Almost 100% of the adult was covered by this study. So after the monitoring period, we got the effective result, and that result is quite it's quite encouraging. We can see basically among these cities, the incidence rate of COVID dropping down significantly, and based on the calculation, we can see the protection rate for the symptomatic incident is 80%, and the effectiveness for the hospital cases is 86%. For deaths is 95%. So basically, so after the large vaccination on this small town, Serena, the people in Serena are already back to the normal life, and this is also quite reported in different journals in the middle of last year. And if you comparing the incident rate, the curve of Serena, comparing with others, the cities around the Serena cities, we also can see significant low of incident rate of COVID in Serena. So that is the first the real world cases by showing the CoronaVac has a quite good effectiveness to COVID. And during that time, actually, the major the major variant of COVID is Gamma P.1 in Brazil, including Serena. So another real-world studies I would like to share today is from Chile. In the May of last year, actually, Chile MOH also released some real-world effectiveness in the whole Chile population. Regarding Chile, actually, the CoronaVac was approved by Chile authorities in the beginning of 2021, and Chile government conduct vaccination campaigns very quickly. From the beginning of February of last year, Chile conduct this vaccination campaign by mainly used CoronaVac. Because at times only CoronaVac, a large amount, several millions doses CoronaVac arrived in Chile. And these studies covered about 10.2 millions of people. The first data released is coming from adult population. It's about more than 10 million population enrolled. And then after that, also there will be several millions of people aged 3 to 16 years involved in these studies. I'm sorry, I think the number in slide is for the people aged 3 to 16 is wrong. Actually, it's not 15. It's just several millions of people. Pediatric people enrolled is also covered by these studies. So generally speaking, 14 days after the second dose of CoronaVac, regarding the adult population, the effectiveness rate for the overall effectiveness is about 65% for elderly, 74% for 65% for adults, 74% for elderly, and 38% for the pediatric. Sorry, I make wrong statement. So the general speaking, the effectiveness against COVID in elder population is 65%, and in the adolescent and some old children, 6 to 16 years old, the effectiveness is 74%. For the hospital cases, adult is about 87%, and adolescent 91%. For the ICU is about 90% for elderly and 93% for adolescent. For the death is only have a data for adult is 86%. Regarding this young kid, 3 to 5 years old, the data is just preprint. I think one day or 2 days ago. So generally speaking, the overall effectiveness against Omicron is 38%, and the hospital cases efficacy about 64, nearly 65% for this younger population, and ICU protection, the effectiveness of ICU hospital cases is nearly 70%. So later I will give more explanation for those numbers. First, let's see the effectiveness for the adult population. So I just mentioned that Chile started a vaccination program since February, And then first they start to the elderly and high risk population, and then whole adult population. And in September, they also used the CoronaVac for the adolescent and some children age. And then December, they are going to use for the younger population, 3 to 5 years old. And this data shows that the effectiveness against COVID in the adult population. The first they published the analysis result in April. It's around 2 months after the whole vaccination program, and then every month they give the update about the effectiveness. And the data published in New England Journal in September last year is coming from the second analysis. That means in May of last year. So you can see the total effectiveness about 66, hospitalized 87, ICU 90. And if you look at the whole data, you can see actually the effectiveness of CoronaVac against serious cases, including hospital cases, ICU, and deaths, always keeping very high level. Even in August, that means that some subject already 6 months after second dose. And regarding the adolescent and children population, just to mention that they first start the vaccination program since the June of last year, but since September, they are going to use the CoronaVac for 16 years, and December 6 last year they start to use the CoronaVac for 3 to 5 years, and so those data was cut off in January and February of this year, respectively. And we can see for the adolescent effectiveness is quite high, is 74% for the general effectiveness, and hospital cases and ICU cases, the effectiveness over 90%, because during that period the Delta is still the domination in the whole Chile population. So that effectiveness also shown that CoronaVac can give a quite good effectiveness for Delta strain in the adolescent population. And the second row is about the data from 3 to 5 years. You can see the number dropping a lot. For the general speaking, for the general effectiveness is only 38%. For the serious cases, still look like okay, 65 to 69%, because that data are coming from December until February of this year. So most of the cases are Omicron. Since January of last year, Omicron already become 90% above domination in Chile population. So
for Dr. Meng for the very updated presentation on the real-world evidence in Chile and very recent information on the Omicron outbreak and with the effectiveness from the in 3 to 5 years old children in Chile, even though the effectiveness is not as high as the adolescent, but it's because of the different strain circulated, and with children is measured at the Omicron. I think that there would because of a lot of information at the time, we have to go to the last speakers, the representative from the Sinopharm, Ms.Meng Li. She's the Director of International Cooperation from China National Biotech Group. She has more than 16 years experience working in the pharmaceutical biological industry, including vaccine. Recently, she organized and discussed with the CRO and relevant authorities to roll out the COVID-19 vaccine phase 3 trial in multiple countries, which play a key role in the deployment of a COVID-19 trial in 6 countries. Now, please, Ms.Meng Li for the presentation.
Thank you, Chair, for the introduction. I'm Li-Meng from CMBG. Today I'll present, I'll representing Sinopharm CMBG and introduce our COVID vaccine, which is an inactivated COVID-19 vaccine. My presentation consists of 5 parts. So I'll start with a general introduction of our company and also our vaccine, then talk about the phase 3 and booster study. At the end, I'll also introduce the data in special populations, including children and people with comorbidities. At the end, will be introductions on our real world studies. I just would like to highlight that now we have more than 10 billion capacity for various kinds of COVID-19 vaccine. And so I'll skip the pictures. For the inactivated vaccine produced with viral cell substrate, it can be stored and transported at 2 to 8 degrees Celsius, which is very user-friendly. And we have 1 to 5 doses well and pre-filled syringes for the supply. The target population approved in more than 100 countries are above 18 years old. And we also obtained EUA in countries like China, UAE, Bahrain, and Argentina for the use in under 18 years old children. So this vaccine has been authorized for use in 118 countries. And up to now, we already supplied more than 1 billion doses to globally. So this is a basic clinical phase 3 design. So I'll skip the details. Now I'll talk about the vaccine efficacy. We have several types of analysis. One is interim analysis, so with a median follow-up time of over 112 days. And for the final analysis, the median follow-up time is 154 days. And we also conducted a durability of protection test with a median follow-up at the time of over 200 days. The total VOC was good. And for the final analysis and the interim analysis, it is 63% and 78% respectively. It also showed good protection in about 60 years old populations. So for the durability study at up to over 200 days, the total VOC can still reach approximately 57%. And for the effectiveness against severe illness is over 90%. And this is the vaccine efficacy in subgroups. This vaccine showed very good efficacy in both female, male, and at different age groups. And it also showed good efficacy in obesity and diabetes populations. This is analysis conducted according to WHO requirements. So we analyze the VE at 60 days, 120 days, 6 months days, and 240 days. So the VE against COVID-19 was approximately 60 to 56% respectively. Until 240 days, it still showed good efficacy post the second dose. So this is the safety result, which showed this vaccine is very tolerated in all populations. The adverse reaction rate in the placebo group is even higher than the vaccine group, which shows this vaccine is very safe for use. And in most of the adverse reactions, it is very mild, mainly grade 1. And the most frequently seen adverse reactions are actually pain at injection site and fatigue. This slide shows the immune persistency up to 9 months is post the second dose. And we can see that the efficacy in 18 to 59 years population is a bit higher than that of above 60, but the GMT level in those vaccine groups were significantly higher than placebo group. And the titer was also tested at those time point post the second dose. This is a phase 3 booster study, and we actually given a booster to people who received the previous primary series 6 months ago. And this study was conducted in a population about 18 years old. And the total subject number is over 9,000. So the participants also includes nationalities from 93 countries. So generally speaking, it showed a very good booster effect. You can see that the total vaccine effectiveness against COVID-19 is 86%. Against severe diseases was 94%. And the vaccine effectiveness after the booster dose was increased by 30% in compared with post-second dose. Which suggests that a booster dose will significantly enhance the immune responses in those participants. And after the booster dose, the antibody level increased by 10-fold. This is the safety results, which showed that the safety profile after the third dose is better than post-second dose, mainly grade 1, and no severe adverse events were found. This part I'll talk about the pediatric populations. This is a study conducted in Henan province in China. It also showed this vaccine is very safe in 3 to 17 years old. The local reaction rate was low, and mainly the most frequently seen adverse reactions are mainly pain at the injection site. This is the immune persistence up to 6 months in children. The post-immunization antibody level in all the younger groups increased with the increase of dosages, and the antibody level at 90 days remain at very high level, which is comparable to 28 days post the second dose, and the antibody level shows some decline at 180 days after the full immunization, which was consistent to the result from the adults. And this is the data from hypertension and diabetes population, which show this vaccine can stimulate good immune response in all those people with comorbidities. Seroconversion rate is 100%. And the vaccine is also very safe to be used in those populations. This is the cross-neutralization result against the various kind of variants. We do see some decline in the neutralizing capacity against the new variants, but it still remains at some certain level, which suggests some protection level to the variants. This is the a study published by Sri Lanka, which shows that this vaccine also has some neutralizing capacity against Delta and Beta. This slide shows cross-neutralizing results against variants including Omicron. And as you can see at the left-hand side, that for the Omicron, there are still remaining neutralizing capacity, and which suggests that it can provide certain level of protection against those variants. So now I'll move to the real-world effectiveness, and this is a quick summary, which are data published by government website and ministries. So we just do a quick summary here. You can see that from different countries, there are data show that this vaccine can provide good protection against both severe diseases and infections. This is a study from Argentina, which was conducted in a population over 60 years old. The sample size is nearly 150,000. And the study lasted for approximately half a year. And the vaccine effectiveness against deaths post second dose was 84% in elders. And this is a study conducted in Peru, which enrolled health workers over 18 years old. This study is a retrospective test-negative design study, observational study. In this study, we enrolled more than 600,000 participants. And the vaccine effectiveness after full immunization against all-cause deaths was 90%, and against COVID-19 cause deaths was 94%. This is a study from Sri Lanka, which suggests that the immunity antibody levels stimulated by vaccine is comparable to natural infection of the B.1.617 strain. And it also showed good protection against the deaths, and also it has good protection against infections in the populations, which is significantly lower than those who was unvaccinated. This is a real-world study from UAE, which is a retrospective cohort study. The population was a PCR positive cohort, and based on those studies, the vaccine effectiveness in fully vaccinated individuals was 80%, 92%, and 97% in preventing COVID-19 related hospitalizations, ICU admissions, and deaths. And this one is an effectiveness study from UAE as well. It shows that the infection rate at 90 days and 120 days after vaccination was 0.72% and 0.97% respectively. And my last slide is about the real-world study in Hungary. This study enrolled people about 16 years old. The number of subjects enrolled was nearly 900,000 who vaccinated with Coilo, and it suggested that the vaccine effectiveness against symptomatic SARS-CoV-2 infection was approximately 70%. So that's all for my presentation. At the end, I'll be happy to take questions. Thank you so much.
Thank you very much, Ms.Meng Li. I think we've got very important and useful information from all 6 vaccine developers right now, especially for the last 2
So next will be
inactivated vaccine developer from Sinovac and Sinopharm, seeing the information regarding the young children from age 3 years of age, and then we see some of the level of protection of hospitalization and severe form of the disease even during the Omicron period of spreading. Right now we come to the session of Q&A, and we have all of the presenters over here as well as the representative of the speakers. So I can start first, Dr. Sunet. We have a number of questions right here, and any of you can address to the question, and you can rest your hand by the icon of hand resting. And right now we also have Dr. Bruce over here in the broadcasting room. So we can start with the question,
Yeah,
please.
I think that we start with the first, a very popular question about the second generation of the vaccine, whether it's being launched as soon. We have heard from several news in the newspaper as well that some companies has announced of the ongoing work on the second generation. So I would urge the speaker whether how each company would like to address about the second generation to cope with the variant of concern.
So
So-
maybe Dr. Carmen, please, if you have any idea on the second generation or the variant-specific vaccine, please.
Yes, maybe I can start, and I think I speak for a lot of us. It's very important to be on top of the situation with the different variants. So several of us, I'm sure, just like our company, Janssen, we're looking into this, and we are looking into developing newer generations of vaccines to address variants of concern. So definitely, our R&D is trying to do some studies regarding this. And let's see how it's very good that several of our platforms have that agility to be able to address this kind of situation. So as new variants emerge, we need to adapt to the situation.
Thank you. Dr. Clarke, I see your hand raising. Please, is there any news from Pfizer?
Yeah, so I'm just going to give the answer on the perspective of Moderna. So Moderna have announced that we have been recruiting for an Omicron-specific formulation, which has been in the clinic since January. And we're also now starting to look at a bivalent formulation containing the original ancestral strain, which we found to be very effective against a number of the variants, but also including Omicron as well. And that study started recruiting this week. So I guess what we're all looking for, is some formulation which is going to give us some broad protection. And that's what we'll be really looking for in the next formulation. But we'll be really driven by the results of our studies to decide which formulation to bring forward.
Yeah, may I add a little bit for this? It's not just a yes or no, but if may you please give the timeline as well, because the people may have heard about the yes, we are developing the variant-specific vaccine, but when it will be available? Maybe I don't know. Yeah, please.
Obviously, we have to be driven by the results of our clinical studies, and we haven't seen the results yet. So, I mean, clearly, we would like to have something available in time for when the next wave occurs. But it really does depend on the outcomes of our clinical studies. So, we need to be cautious about making promises.
Thanks, Dr. Bruce, may you?
Yeah, it's a very interesting topic, this one. And I mean, as you know, AstraZeneca have been working towards a beta variant vaccine. We kind of use that as a pilot to demonstrate that the technology is there to plug and play with different variant spike proteins. Obviously, the results from that study have read out quite recently, not formally published, but we did announce that the results, particularly in terms of benefit for Omicron, were not significantly better than we're seeing with the current vaccine anyway. So we've actually made the decision not to progress with the beta variant vaccine. But I think the same is true for an Omicron-specific vaccine. I think given the timelines and how dynamically things are changing, by the time these vaccines come to market, the wave will have passed and there'll be another variant. So I think to Chris's point earlier, the holy grail we're looking for, I think, is a variant-independent vaccine approach, a pan-serotype approach. I'm not sure anybody's there yet, but as the pandemic evolves and new variants kind of come and go, I think that will need to be the approach that's looked at, something that works across variants rather than focusing on specific variant vaccines. It's a challenging from an R&D perspective to do that quickly enough, given how rapidly the evolution is happening.
Mm-hmm. Thank you. May we have the additional comment from the other speaker, please? Dr. Egemen, or
Yes,
please.
I can say a few things for Pfizer. So it was already announced in January that Pfizer is also working for an Omicron-specific vaccine. But this is a multi-arm study comparing also the fourth dose of the current one with the new Omicron one. So we will see the results, I hope, in a couple of months.
Okay. Anything else from our colleagues from the Sinovac and CNBG, please?
Okay, so I also would like to provide some update from CNBG side. Actually, at the beginning of the vaccine development, we explored almost all kinds of technical platforms. We have all those technologies in-house. And finally, we decided to use inactivated vaccine. It is mainly because that this vaccine was tested extensively in the past decades. But we also have a candidate which was produced with recombinant platform as well as mRNA platform. So for the second generation inactivated vaccine, the Omicron strain inactivated vaccine is already available for the clinical trials. So we are now preparing for the clinical trials of the vaccine, and the vaccine has already been produced in large scale. And for the recombinant vaccine, in the past year, we actually developed the recombinant vaccine against the prototype and also against the alpha, beta, and delta trimer recombinant. And we have already obtained the emergency use for those two recombinant vaccine in UAE. And we also have already completed the development of Omicron strain recombinant vaccine, which is now also waiting for the clinical trial. And for the mRNA, it is at preclinical stage. So for the later on, we will conduct further research, and it will also be available for the clinical research in the coming months. So that's the update from our side. Thank you.
Yeah. Thank you very much. I think that this question we have to answer very often, but the lay public have to understand whether first about the vaccinology concept, whether that seed that in development is immunogenic enough, is that the correct seed that can, when we provide as a booster, it can cause protection to any other variant of concern previously as well. And second point that about the timeline, the lead time for the clinical development until we can prove that the new generation or the new variant of concern vaccine is working well. So seeming at from all manufacturers, the current vaccine one that we have used currently is still working well when we provide as a booster, right? So I would come to the next question that people are asking about. Is there any correlation on antibody titers and effectiveness in the real-world studies? I have seen some of the slides that Dr. Bruce has presented, compare the immunogenicity on one side and the effectiveness on the right side. Would you like to comment something on this?
Yeah, so I think there's a lot of misperception, I think, going on about what these vaccines can or should be capable of. Correlates of protection are nice, and it's kind of a holy grail for vaccine development that shortens the process for new developments to come in quickly. But obviously for coronaviruses, it's not that simple. You know, we tried initially with our clinical data to look at correlates of protection against symptomatic, overall infection. And there is a bit of a trend when you look at that. If you do the same type of analysis for asymptomatic infections, there's no correlation at all. And obviously from the data I showed as well, for more severe outcomes, there's no correlation whatsoever. So we need to be a bit careful, I think, in terms of how we're interpreting these antibody studies. It's easy to measure antibodies, and that's why people do it. It's quick and it's fast, and you get a nice result. But we need to be careful about how we're interpreting that, I think, in terms of what we expect from these vaccines.
Any other comments from the other speaker, please?
Dr. Carmen.
Maybe,
Yeah.
yeah, please go ahead.
Yes, just to add that there are different endpoints in mind. Remember that we have to look for protection against infection or protection against hospitalization or against severe outcomes like death or ICU admissions. So if we're looking for that holy grail of correlative protection, those different endpoints might be at different levels. And then there's the interplay also of neutralizing antibodies and of cellular immunity. I think both have their roles to play, especially against severe infections. We see a lot more data coming out on cellular immunity and the role of cellular immunity against this endpoint.
Mm-hmm. Yes. So, anything else, Dr. Egemen?
I just want to add to Dr. Carmen's comments. Oh, sorry.
Please. So, lady first.
Yes,
Sorry to interrupt you. Sorry, I didn't notice that there is some delay for the networking. And so I think it is very important to keep on monitoring the effectiveness against the different various levels of COVID-19 cases, including infections and also severe cases, as Carmen just mentioned. And for us, we are also doing some analysis to show these correlations. And if there's any update, we will provide that to the public quickly. But according to our current observation in some studies, it shows that the infections in terms of vaccine effectiveness against the infections, after the third dose, there is no significant differences in terms of infections in the group who received the activated vaccine or received mRNA vaccines. So we didn't see significant differences among those groups. So this is just a quick update. But anyway, those data later on will be published, and we will keep the public informed if there's any news.
Dr. Egemen.
Yes.
Yeah, very quickly. I think we are still learning. There is a lot that we don't know about disinfection and vaccine behavior. So we need to keep in mind. Of course, antibody levels are critical and important and one of the maybe only parameters that we can measure for the new variants. However, it's not the only parameters. So cellular response, I can agree more, is very critical. We have to keep in mind the T lymphocyte response is also very critical in long-term COVID. So we are still learning, and there are more parameters we need to keep in mind, and we don't have still a correlative protection.
Thank you. Dr. Clarke, please.
I was just going to say something very similar in that I think that actually antibodies actually have been quite useful, particularly looking at cross-neutralization between strains. And there is, while there's certainly not a correlative protection, but we do see some kind of a link between waning neutralization activity and waning protection against infection. But there really are a lot of other things going on in the immune response to our vaccines, which we need to look into more closely.
Thank you. I think in the short message to the audience that antibody level is only one of the proxy indicator only, and we do need to measure the other indicator as well. And the more important is the effectiveness result that we have seen today. That even the waning of antibody level, but the effectiveness will still be there, still be remained, meaning that there are more indicators that we did not measure it or we did not demonstrate it. And the people and the developer is looking at the correlative protection level, but not yet for right now. So we do need to rely on the effectiveness result as well. Okay, maybe we have the last question, but very important.
Yeah, I think that it would be a question from the lay public or our user that can you, what do you think, how often do we have to get the booster? Is that would be every 3 to 6 months or a year? What do you think?
So that's the million-dollar question at the moment, isn't it? So I think we're still early. I mean, I know I know people are sick of the pandemic and need it to be over, but we are still relatively early. We're talking about 12 months experience with most of these vaccines. So that long-term duration of protection data is still developing. I think it's encouraging that we're seeing quite durable protection against severe outcomes, hospitalization and mortality. That's likely to be much longer. In an Omicron world, clearly protection against infection is waning quickly, and I think that's that's a challenge. But at the end of the day, as long as we're preventing people from being hospitalized and preventing them from dying, the vaccines are still doing what they're designed to do. So I think provided we're seeing good duration of protection with those numbers, we don't need to think about boosting every 3 months or 6 months perhaps. I think there's obviously a longer term plan that we need to consider.
I hope so. Please. If there is an additional to this, Dr. Clarke, please.
Dr. Clarke.
Oh. Yeah, look, I didn't have my hand up.
I
But
see. I see. So, meaning that we have the same answer like Dr. Bruce, right? Because we don't know
very
yet.
similar.
Okay.
Yeah,
Very similar
hopefully that
position.
we can have
Annual.
the annual
Annual.
vaccination for the people to prevent hospitalization,
Yeah.
severity, and deaths. So that the aiming for the world. I think it's time. To me, I'm realized that 2 hours of discussion is not enough. We may need 2 more hours, but the speaker or the audience may be exhausted before that time. So thank you very much for all speakers, Dr. Carmen, Dr. Clarke, Dr. Egemen, the representative from Sinovac, and Dr. Li Meng, and also Dr. Bruce over here, with us in the broadcasting room. We are very delighted and grateful for your contribution today. And I would like to close the session. Thank you very much, and we can work more on this endurance fighting with the COVID-19. Thank you very much.
So we will throw this to now the stage to the MC, Kun Waravit, for the next session.
Yeah, that's it all for today meeting ครับ Our keynote speakers deliver a fantastic presentation. I hope attendees took away a wealth of knowledge and experiences that inspire our interest in vaccine R&D as well as a greater grasp of the unique technological platform built by our global partner ครับ On behalf of National Vaccine Institute and the conference organizing team, I would like to express my deep gratitude to Dr. Nakhon and Dr. Sunet, as well as our honorable speakers for their insightful presentation at the conference. Your contributions will assist us to greatly expand our vaccine competence. We certainly gain a valuable insight from your perspective. Moreover, I want to express my appreciation to everyone who took the time to attend this session. Following the session, all attendees are asked to complete a questionnaire. You can participate in the survey by scanning the QR code and by clicking the link at the bottom of your live streaming page. Please be informed that all of your feedback, comment, and input will help us innovate and succeed in the future conference. If a mistake happens, we NVI team apologize to everyone for any inconvenience caused by this occasion ครับ So currently we have another oral presentation in another room, and the session of the preparedness vaccine development and production for emerging infectious disease control in Thailand, part 2 of the team Thailand. We already conduct this meeting topic at the conference room the V room live streaming. We're waiting for you to participate. Please enjoy your break and come and go see through all the instruction video clip that we provide by us. This NVI end room will close for today and for another day. But still, we still have we and I room live streaming for you to participate in. Thank you so much for joining us by today's session. And we now close the end room for today ครับ Thank you